Okay, so we're back with part 2 of chapter 20, the digestive system, and we're going to talk about chemical digestion of nutrients. So how macronutrients are broken from larger molecules down into smaller molecules and absorbed in your bloodstream. So breaking these larger molecules down into smaller ones is called catabolism. And this is aided by the use of enzymes. Enzymes are proteins that are created and secreted in the salivary glands, in the stomach, gastric pits, as well as in the pancreas and pancreatic enzymes. And these enzymes help break bigger molecules down into smaller molecules or catabolize them. And this is also using water to break these molecules down. And so they're hydraulic reactions. We're using water to also break them down. Nutrients are absorbed in the small intestines, and all food stuff is absorbed except some water and indigestible food materials, cellulose for one. 20% of electrolytes like sodium, potassium, magnesium is absorbed. Most water is going to be absorbed because it follows solutes. So that's going to be dependent on how much sodium is in the food stuff and so forth. So materials pass across the membrane into the interstitial fluid of the submucosal layer and then into the bloodstream, the mesenteric capillaries, or in some cases, fats go into the lymphatic lacteals or small capillaries. So we're going to talk about specifically how carbohydrates do this process, how proteins and how fats do this process. So carbohydrates, we consume them as polysaccharides or disaccharides. Now, disaccharides are what we call simple sugars, and polysaccharides are what we call complex carbohydrates. So polysaccharides are complex carbohydrates, disaccharides are simple sugars. And so examples of disaccharides are sucrose, lactose, maltose. So this is right here, this is sucrose. It has a fructose molecule and a glucose molecule. Starch, glycogen, and cellulose, those are all complex carbohydrates or polysaccharides. And they can be hundreds of molecules long, bound up together. So you can have starch and cellulose all bound up together. Starch, we call it polysaccharide starch because we have enzymes to break down starch. So have enzymes to separate glucose. And cellulose, we call fiber, do not have enzymes to break down glucose. Only monosaccharides are absorbed by the intestinal cells into the body. So everything has to be broken down into a single molecule. So all these need to be separated out to be absorbed. And so where that breakdown starts occurring, again, salivary amylase in saliva starts breaking down parts of the bonds of carbohydrates. Now, if they're simple sugars, it's not allowed to break down. But that salivary amylase is working all the way up to the small intestines. Then in the small intestines, you get pancreatic amylase released from the pancreas. And the brush border enzymes are the final enzymes. And these brush border enzymes reside here on the microvilli. And so the microvilli is an extension of the cell membrane, but it also contains enzymes, lactase, maltase, and sucrase These are the brush border enzymes that break down disaccharides. And so carbohydrate digestion is a process. The enzymes involved are salivary amylase and pancreatic amylase. The end product of salivary amylase and pancreatic amylase is small disaccharides or limit dextrins. These small disaccharides, again, they're simple sugars or simple carbohydrates, and limit dextrins are really just a few glucose molecules together. These enzymes are going to be on the brush border or the microvilli. And so dextrinase breaks down limited dextrins into glucose. Glucoamylase is going to break down polysaccharides into glucose. If it's table sugar or sucrose, it's going to be broken down by sucrase. If it's milk sugar or lactose, it's going to be broken down by lactase. If it's malt sugar or maltose, it's going to be broken down by maltase. And these enzymes are on the microvilli or the brush border. Now, once these brush border enzymes break them down into single molecules, so this right here is showing you, these right here are the villi. I'm sorry, microvilli. And you have a brush border enzyme here, maltase. And so you have the disaccharide maltose. This enzyme breaks it into two glucoses. And then glucose is secondary active transport across apical membrane of the epithelial cell or the absorptive cell of small intestine. So if you guys recall secondary active transport, it relies on primary, well, that's not dark enough. Let's do this. Primary active transport. So we have a sodium potassium pump on the basolateral membrane of the absorptive cell, and it is pumping sodium out of the cell. Sodium's leaving, it creates a concentration gradient. So sodium that's inside the small intestine can then go passively down its concentration gradient that was created by actively pumping sodium out of that cell. It's going to have a co-transport with glucose. And so glucose is said to have secondary active transport because you have to have active transport of sodium to create the ability for sodium to go down its concentration gradient and bring glucose into the cell with it. Now, once glucose is in, it's basically facilitated diffusion that requires no energy because you have a high concentration of glucose now and a lower one as it leaves. And so then glucose passively through facilitated diffusion goes in the interstitial fluid, and then that facilitated passive diffusion goes into the bloodstream. Fructose, on the other hand, so that's for glucose. Fructose is facilitated diffusion both at the apical here and here. And I really should change color. So here, facilitated diffusion on both sides. So, you know, all calories are not the same. If they are disaccharides or sugar, it's one bond that the brush border enzymes break down and then absorb. If it is, and we love fat, we love salt and sugar together, right? Salted caramel, salted everything. And so that salt also helps transport that sugar right across that absorptive cell. If it is a polysaccharide that has starch and cellulose, that we call fiber, it takes a whole lot longer to separate out the glucose molecules from the non-digestible cellulose. And so that breakdown is much slower. The glucose enters the bloodstream a lot slower. And so it has a lower entry of glucose, therefore not really having an increase too far above what is homeostatic for glucose levels in your blood or blood sugar. And so that allows the glucose to go in your blood and just naturally go facilitated diffusion into your blood. Now, you have these transporters, these Glut4, they're called Glut4 transporters. And some of them exist in the cell membrane. Insulin, when insulin gets released, it increases the number of these, right? So you can have more glucose going into the cell. So insulin promotes more of these Glut4 channels to help facilitate glucose going into the cell. Now, if the cell needs the glucose, it's going to use it to create ATP. It's going to use the energy in the glucose and transfer it into ATP. And then it can use it for whatever's going on. Or if it's liver or muscle, skeletal muscle, it'll store it as glycogen. But there's a limit to how much glucose you can store as glycogen in the liver and in the skeletal muscles. If you have an excess that you can't store, it then gets converted and stored as triglycerides in fat cells. Too many amino acids can also end up stored in fat. So calories are not everything. It's how quickly that glucose can be separated into individual molecules and enter your bloodstream. And if you have a huge insulin release, insulin promotes storage. So the more insulin that gets secreted, the more you're going to store everything that you absorbed at that point in time. And so again, the bigger the increase in blood glucose levels, it's going to cause more insulin to be released. More insulin is released, more storage of these nutrients. So 100 calories, depending on the food you ate, is not necessarily 100 calories to your body, right? Because you're going to spend energy separating the fructose and glucose out of apples. And so you're spending energy to get out energy. And so you don't get a whole 100 calories of potential energy because you use some of it. But if it's sugar, right? If it's sucrose, 100% of that sucrose is going to enter your bloodstream and you will get all of that energy. It takes nothing. And so foods are rated on what's called the glycemic index. The glycemic index rates how quickly a food can get, or how quickly a glucose can get into your bloodstream from that food. So you only have really the glycemic index for carbohydrates, but it's a measurement of the presence of glucose in the blood after the food has been ingested. So how quickly is it broken down and how quickly does that glucose get absorbed in your bloodstream? So high glycemic foods, the glucose enters your bloodstream very quickly, causing insulin to have to be released. And so diabetics use this glycemic index to decide how much insulin to inject. That would be type 1 diabetics. Type 2 diabetics, they still do produce insulin, generally speaking, but the insulin no longer works. So, but they could still use the glycemic index because no, it's not good to have blood sugar levels rise drastically after you eat food. It's just not good for your body. And you end up storing more of that energy as fat than a low glycemic index food. And so you can look up glycemic index. It kind of shows you different foods and how they're rated. So it's helpful. And so those are a couple links. So to kind of wrap it up, this diagram kind of shows you if it's carbohydrates, right? It's complex and simple sugars, which are starch and disaccharides, salivary amylase and pancreatic amylase, are the enzymes that get released, and they break things down into small chains, oligosaccharides or disaccharides. Then the brush border enzymes are going to break that reside on the brush border, which are here, are going to break it down into monomers or monosaccharides, single molecules, and those are going to get absorbed. Now, you could see site of action and then path of absorption. Glucose and galactose are secondary active transport. So this right here is secondary active transport. Fructose is facilitated diffusion. Facilitated diffusion on the basement membrane. Now, fructose is facilitated diffusion, both this apical surface and the basolateral surface here, right? And so one reason fructose is kind of good, if you're doing endurance activities, having some fructose is nice because you don't necessarily need secondary active transport. It's pure facilitated diffusion. So if you have an influx of fructose, they use fructose in a lot of those goos for endurance stuff. Too much fructose, it doesn't all get absorbed because it's facilitated diffusion and you're kind of relying on how many facilitator molecules. So too much fructose, you're going to end up with that fructose staying in your small intestine with water and you can get crampy, diarrhea-y, but a small amount of fructose, it's going to get into your bloodstream quickly without having to actively transport it and use energy. So you might see fructose and put into things. Okay, so proteins, they are broken down into amino acids, and the enzymes that break down proteins are called proteases. And there's endopeptidases, exopeptidases, and zymogens. Zymogens are the more common ones. Zymogens are usually stored in the inactive form because you don't want an enzyme that breaks down proteins kind of roaming around unregulated. So there's usually something that will activate zymogen proteases. They're secreted by exocytosis out of the cell. So for example, the chief cells, and then activated by something like hydrochloric acid that'll clip or lyse something off that enzyme or that protease. So that's proteolysis, and activate the enzyme. So proteins, again, get broken down in the stomach. They get denatured, and pepsinogen, which is then activated into pepsin, which is a protease, will then cut it into smaller peptides. So this is showing you the stomach right here. And then these smaller chains will go into the small intestine, and the pancreatic proteases will then break them down into trypsin, chymotrypsin, and carbopeptidase, or into these smaller forms. And then these brush border enzymes can then break them down into single amino acids. So you're looking at a protein. It was denatured in the stomach. It's now in the lumen of the small intestines. You have the brush border enzymes here that are breaking each amino acid off, and it's getting secondary active transport with sodium and then facilitated diffusion on the basolateral surface for absorption. So this goes through protein breakdown. And again, when you look at it here, by the way, it's active transport, secondary active transport, and then passive transport, facilitated diffusion. If you look at this diagram, enzymes, pepsin in the stomach, which is activated by the presence of hydrochloric acid, and then pancreatic proteases, trypsin, chymotrypsin, carboxypeptidase, and then brush border enzymes, aminopeptidase, carboxypeptidase, and dipeptidase. Those are the brush border enzymes that break them down into single amino acids, which then can secondary active transport with sodium, and then facilitated diffusion on the basolateral surfaces. Here. Some di and tripeptides can be absorbed through co-transport with protons and hydrolyzed amino acids, but this is the bulk of how they get absorbed. Lipids. Now, lipids are hydrophobic, but lipophilic. And so they can't exist really in water, but they can go through cell membranes. And so they pose a little bit different problem, and that's where bile salts also come into play. So the enzymes that digest fats are called lipases, and the pancreas is the only real secretor of lipases. But lipases can only get to the molecules on the outside of these fat globules. And so bile salts, which are secreted by the liver and gallbladder, starting to lose it now. Okay, let's try that again, shall we? Liver, gallbladder, secrete bile salts. And so bile salts are going to increase the surface area of the droplets by breaking each into a smaller droplets called emulsification. So what the bile does is the bile salts kind of attack this big glob. The bile salts cling on and make the fat droplet become a more polar molecule, so it can exist in a polar environment. And so the bile salts are on the outside, they're polar, and the smaller fat molecules are on the inside with the phospholipids and the triglycerides. So bile salts break down a big glob of fat into smaller ones. So now the lipases can really get at these little molecules. Again, bile is coming from the liver and the gallbladder. It's secreted into the duodenum or duodenum. It then will break down these big chunks of fat with lipases into kind of amphipathic, so they can then be absorbed into the mesenteric capillaries and then go through the hepatic portal vein. And then guess what? The liver gets it right back to make lipoproteins like HDLs, LDLs, cholesterol, anything that will carry stuff in the blood. So in the lumen of the intestines, right, these bile salts make these fat droplets that can have triglycerides. You can get lipases from the pancreas that then break these phospholipids down into fatty acids or monoglycerides. It can also package them into what are called micelles, if that's a phospholipid with some triglycerides. And then these fats can get absorbed across the cell membrane because they don't need a transporter since they're fat-based. So they're absorbed across. Once they're in the enterocytes, they get reconstituted either into triglycerides or packaged into what are called chylomicrons. So fat clusters with bile salts form micelles, and these micelles can diffuse into the epithelial cell and combine with proteins to form chylomicrons, so it becomes amphipathic. And these are going to enter a lacteal. Now, this is a lymphatic capillary. And the lymphatic capillary then will bring these chylomicrons and carry it all the way until it gets to the subclavian veins, and then dump the lymph into the subclavian vein along with these chylomicrons. So that's how the chylomicrons make it into general circulation. The other way to absorb fats is you absorb them, well, this goes through the one I just went. You can also absorb them straight across the cell, and it can then enter the bloodstream. So I believe there is a second. I thought there. So fatty acids can also just get absorbed into the blood as well as triglycerides or other lipids. So these triglycerides can also just enter your bloodstream. So not everything is traveling in the lacteals. That's what I'm trying to tell you. So emulsification of fat begins with bile being secreted from the liver and gallbladder, and then pancreatic lipases. And this does not start to occur until the small intestines. And this goes through, enters the intestine cells via diffusion. They form chylomicrons that then can go into the lacteals and lymphatic system, which then gets dumped into the thoracic duct, subclavian, left subclavian vein. But some short chain fatty acids can be absorbed into the blood and then go to the liver via the hepatic portal vein. Vitamins, fat-soluble vitamins have to be absorbed with lipids in these cells and chylomicrons, but water-soluble vitamins just require transport proteins. Again, vitamin B12 requires intrinsic factor from the parietal cells in the stomach. Sodium chloride, potassium, calcium, some of these are absorbed hormonally or just through facilitated diffusion. This goes through iron and water. By the way, anything, malabsorption is the inability to absorb the nutrients from your small intestine into the blood. And so anything that interferes with breaking down the macronutrients into single molecules is going to cause malabsorption. And so also anything that interferes with the delivery of bile or pancreatic enzymes can result in malabsorption, or things not getting broken down to single molecules and getting absorbed. And so that's, celiac disease, anything that disables this breakdown and process. So that's chapter 20. Chapter 21, we'll talk about absorptive state, post-absorptive state, and we'll wrap it up.